So, good morning. My name is Jeffrey Robens, and I'm a JSPS Research Fellow working in Dr. Mineko Kanagaku's laboratory here at iCeMS. And the iCeMS Crosstalk series presents two iCeMS scientists together in a discussion format. Today I have the pleasure of speaking with Dr. Koichiro Tanaka, who is the head of the Terahertz Optical Science Laboratory here at iCeMS, and I believe has the unique distinction of being the sole solid-state physicist at iCeMS as well. So good morning, Dr. Tanaka.
Good morning.
So I think we all have our own personal reasons what got us interested in science. What is it about science that first attracted you? And about how old were you when you decided you actually wanted to become a scientist?
Yeah, it was at 16 years old. When I entered high school, I joined a biology club because I like nature things.
Sure.
And I started to study biology. Another important feature of our biology club is previous members, high school graduates, taught me many things. How to culture cells and how to culture many small insects. And also they taught many books. One of important book they taught me is a book written by Nico Tinbergen. Do you know Nico Tinbergen? Who is a Nobel Prize winner in medical science. And his specialty is animal socialism. I decided to enter Kyoto University because Kyoto University is one of the center of zoology, and at the time, Professor Hidaka has some leading group, and I want to join the group. So I chose the Kyoto University, and I entered Faculty of Science. Many of Faculty of Science students are interested in physics because the Faculty of Science is famous for theoretical physics.
Okay, I see.
At the time, only the Nobel Prize winner in science field was Professor Yukawa and Tomonaga, who was graduated in Kyoto University.
I see.
So many students who wanted to study physics are coming. So my friends, of course, want to study physics.
Okay.
So we just started to read physics book. After 1 year passed, I forgot the initial motivation, zoology, and was enthralled by physics.
I see.
Because after learning university physics, I was attracted by simplicity and also some strict logical structure in physics. And I love such kind of things. So I decided and changed my mind to go physics field and to be a physics scientist.
I see.
So I'm sorry, it's a long story.
Oh, no.
But initially, I like nature. That's the reason why I want to be a scientist.
Okay, very interesting. Now, for someone who's not a physicist, how would you actually describe your work in terahertz optics? Like, what types of goals is your lab hoping to accomplish within this area?
Yeah. So I have two subjects. One is typical physics study using semiconductor. It's a nonlinear terahertz optics to study semiconductors.
Okay.
So the goal is to developing a transistor working in high frequency field, terahertz region. So there is no such a nonlinear device existing at the moment. So we want to have a material or a new principle working in terahertz frequency region.
Okay.
So my PhD students in Department of Physics working hardly such kind of things, topics.
Mm.
The other topic is developing optics elements, especially there is near field microscope. So maybe you know, microscope has some spatial resolution limited by diffraction phenomena. And the refraction limit is related to the wavelength. And the wavelength of terahertz wave is around 300 micron. So it's quite long to see details of materials and also details of cells. To see details of cell material, cells, we need much more spatial resolution. So we are now developing a near field microscope, which spatial resolution is around 1 micron.
Oh.
And for the biologist, I must say one thing. Terahertz wave is quite sensitive to water and proteins and many big molecules. And also quite sensitive to the change of temperature and also some mixture contents. So if we develop a terahertz microscope and see the cell inside cell, maybe we can see some fluctuation of temperature inside cell or some movement of proteins. So there is no such kind of microscope without labeling, monitoring such kind of change. So I hope we will serve such kind of observation device to cell biologists and want to collaborate with them. That is a reason why I joined iCeMS.
So being one of the few physicists at iCeMS, and as I mentioned, I think the only solid-state physicist here, what types of collaborations are you hoping to get involved in with the different scientists here?
Yes, I have now collaborating with two groups. One is chemist, and like Professor Kitagawa and other excellent chemists are developing new materials, including some self-organized proteins and DNAs. So one of the most important topic is some characterization of newly developed material using physical technique. But this is one collaboration work. The other is so by constructing our new microscope, I want to apply, I want to serve our microscope cell biologist. And try to start some new things. For example, my microscope may see density fluctuation of waters. And maybe we can catch, hopefully, cell division process before visible camera can see.
Because you can already see inside the cell the different fluctuations that are occurring.
So I want to study such kind of biological collaboration work.
Yeah, I think for me that's actually quite interesting because with a normal visible microscope, you're limited by basically what you can see. So you're limited by basically the ultrastructure of the cell. You can't really see what's happening inside. If you do want to see inside, you have to use different types of fluorescent markers or other more invasive type of techniques. But with your microscope, you're actually able to go inside the cell without perturbing the native environment of the cell and see things.
It's basically a label-free technique.
Oh, very nice. Sounds very interesting. Very good.
I'm a physicist and very few physicists in iCeMS. So is there anything you expect to physicists in some collaboration work?
Well, what I hope, because I feel that in some areas, in terms of cell biology and kind of the microscopes, because we do most of our stuff is based on microscopy, there's a lot of limitations. And so I think those limitations then put limitations on our research as well. So that's why I'm interested in particularly the near terahertz optical microscope.
Yep.
It gives you a chance to expand on what you can normally study in cell biology. So for me, those are the kind of things that I'm particularly interested in with your work.
Okay. So I hope we will start collaboration work in the future.
I hope so. I hope so. So as a successful scientist yourself, what types of suggestions or advice would you give to younger scientists who are aspiring to succeed in science today?
Just say, have a dream and concentrate it. And also have a hobby to keep balance. And only the research works is not so good. So both sides important, and every weekend we must refresh. Not only weekend, every day, I don't know. But concentration means that every day I must consider the research works.
Sure, sure. Well, thank you very much, Dr. Tanaka. It was a pleasure speaking with you today.
Yeah, you're welcome.
Thank you very much.
Thank you very much.
Well, thank you very much for watching the iCeMS Crosstalk series with Dr. Tanaka, and hope you enjoyed it. Thank you very much.